you very much. We are Endure Biotherapeutics, San Diego, California. And the forward goes forward. So synthetic biology is a very hot topic right now. And it's a very exciting area, and we're happy to be a part of that. Okay. But the promise is resident bacteria that can make a functional protein with a signal in the body. Okay, that's the key here. But the problem is the current therapeutics under development, they don't remain in the body. They can't endure the gut lumen. They don't survive. Okay, so what we have is a platform to develop biotherapeutics that engraft in the gut. This has not been done previously. So our solution basically is to take native bacteria from a patient. This could be autologous, this could be allogenic, and we knock in a gene of interest into that bacteria to give it back to the patient. Could be autologous, could be allogenic, and it will remain in the patient, and that's the key. So here's a paper that will be published in Cell. It's just been accepted to Cell. And what you find in the graph on the left is colonization in this is mouse for the life of the animal. So generally speaking, what we would compare against would be a day, 2 days maybe, and this is the life of the animal. This is done hundreds of times, colonizing the life of the animal. What's also important is where do you colonize? So we colonize much more than just in the colon. We colonize in the proximal gut. If you have a metabolism play, you need to do that. And finally, here is a bio readout, 15 weeks. This is an OB mice. Looking at diabetes, but 15 weeks after one treatment, you still see a statistically significant response in glucose tolerance. 15 weeks, one administration, pretty cool. So internally, we have an orphan drug focus. We intend to do these in-house with our clinical development. Our first program, however, has gotten a lot of interest already in PKU. But we want to partner the larger ones. So these large indications, diabetes, obesity, type 2 diabetes, this kind of thing, we'll partner with larger companies. Here's our plan, proof of concept, 3 key studies to validate the platform. This is a $3 million to $5 million raise. We're going to do that PKU study in mouse. We're going to validate in the pig or the monkey, and then a couple safety studies. And that will validate the platform. The following raise would be to play out some of those therapeutics. Pretty straightforward. We've just been accepted to Cell, so this we published in the coming months. So finally, we are a platform therapeutics company with an engraftment technology, potentially curative solution to some of these conditions as opposed to a chronic therapy. We have a 3 to 5 million raise to just play out that platform. But most importantly, we already have an early stage partner for the first play here. And that partner is the #1 in PKU development therapy. That's an orphan drug company. They want to partner with this because this is a great solution. Thank you.
So unfortunately, like the microbiome synthetic biology companies have been kind of a graveyard for the past year or so due to this engraftment issue.
Correct.
It's awesome that you're trying to solve this problem. Very glad to hear it. Would you mind speaking a little bit about the engraftment technology itself?
Yeah. So, and you probably know, scientifically, we're 10 to 20 years out of fully understanding how to engraft. Okay, so what we're doing, instead of engineering NISL, which all of our competitors are using, NISL came from 1917, World War I. World War I, NISL 1917, NISL, World War I. We're just taking native bacteria found in man. It still has its antimicrobial peptides, its quorum sensing factors. We're engineering the parts of it that don't affect that, and it can remain, it can survive, and that's what we're showing here. Now, we need to play that out in man, understandably, right? But that's the difference here. And it's different to genetically engineer that E. coli, but everyone should know that 30% of biopharma products are made in E. coli today, 30% of the approved ones. Okay. But those synthetic biology companies have used mostly NISL, just doesn't engraft. So we're using native strains that can engraft. It's a platform. As we develop each one, we tend to patent each one, right? Composition of matter of each engineered microbe. Thank you. Good question.
Can you speak a little bit about why PKU first and your indication selection?
Right. So we're interested in inborn errors of metabolism, so we could knock in one gene into that and solve that problem. PKU is one of the largest indications in orphan drug. We also have the #1 company in that has had 3 huge plays, maybe I don't have to mention them, in that area who wants to partner with that because it's a great solution. So we think it's a good opportunity. It's one of the biggest orphans, and we can do that part in-house, partner it or not. It'd be a great partner, so I hope to announce that soon. But we're early stage, guys. We're seed. We do need this funding. Partner needs us to do our part of the bargain here, as every partner has to do. I had to slow that program down already.
Who's the partner again? Oh, sorry.
I'm not saying that it's
No,
under
you
CDA.
mentioned- you mentioned a partner earlier. I thought you mentioned.
Nope.
Okay. You never mentioned.
What I said was they are the largest in PKU development.
Okay, right.
They're pretty well known. Because of that, I could answer that privately, but we have a CDA, so I'm trying to be honest.
There's targets out there. Let's just say that.
Can you
Where, Dr. Wilson, where are you in the process of raising the 3 to 5?
So we've got friends and family lined up, and that's it. We're just starting. I'm the first person to do this. We're spinning out of the UC, so we're UC San Diego. We're going into private lab, maybe the last place I started. Thank you for asking. What do I do? I founded a company called Matrices Bioscience, which is a skin microbiome company, preeminent skin microbiome company. I have 30 years in biopharma. I made the deal for Halozyme, $612 million deal that made them a $5 billion company. I was the 5th employee there. More of a BD business guy as an engineer, and I'm an engineer. Okay, so this is synthetic biology company.
Can you give any details to the nature of that partnership, the terms, or just like
No, it's
overall?
early. No, it's early. I can't say it's a partnership. I'm saying that we've got really strong interest, and I have to do my piece of it. They're willing to run our compounds right now, and we need to do a little work on my side. Okay. I don't want to oversell that, but that partner is very, very interested, and we're meeting Monday. Everybody should know that Bio is next week, San Diego.
Who are you looking to raise? I guess what's your strategy on fundraising from? Who are you looking to raise from?
So the proof of concept here is just playing out that, right? 3 to 5 million. After that, we're going to go 20 million and start playing out those therapeutic plays. Now, we may get a partner here. We may not.
If I can rephrase
I didn't answer that well.
If I can rephrase that, are you looking for angels for the 3 million? Are you looking for VCs with small checks? Or who are you looking for the 3 million from?
Preferably a seed venture group, okay, that could continue to support us. We know that a lot of the groups don't want to go into an early stage thing where there's 3 million because you have to do all the work to clear that. But we could keep going, and that's what we'd prefer to do. We've got angels lined up, but you know, this 3 to 5 million. So we need a little bit more than that.
How much
probably.
money do you have lined up, if you can say? How much money do you have
A
lined
little
up?
less than $1 million.
Okay, so
We have some legitimate investors in the Bay Area. I don't want to oversell that either, but that we've done due diligence. We've got other synthetic biology professors interested in us as well. Stanford has a very strong program. So
has anyone priced it yet? Oh, I'm sorry. Go.
no.
Can you talk a bit more about the team today?
Right. This is the team. So we, I said I'm seed, right? We're seed stage company. So we have a website now, all right? So we have UCSD. We have a whole laboratory at UCSD that has $5 million in funding, but we're spinning out the company. So that's what I'm doing. We're part of that raise is to bring in 3 bacterial engineers and to optimize those constructs, okay, locally, and that's what we're going to do. So we're a small team. We're seed raise, and that's what we're going to do. Yeah. Thank you. Good question. Great question.
Maybe to that point, are you going to need a clean vivarium for these studies, or
No.
have access to one?
No. If you do things in a clean vivarium, you're going to get those kind of results, and you have to do it really in the real world. So but the professor has done these in clean as well as dirty, and it really has to be a dirtier facility. So, you know, to mimic life. But people don't really understand very well the lab, the microbiome of a lab animal. It's not well understood, so it's not something that people publish on. We need to do real pig, that kind of thing.
So you don't need access to anything special. You can just run it.
No, these will be BSL-2 in the beginning until they're proven, just as any construct would be. Yeah. Nothing too novel in that.
I think.
So, it's a partnering scenario, and that's kind of what my background is. And so we would identify a partner that would fund the subsequent clinical trials, and we'd get a royalty and milestones from that, generally speaking, that kind of a deal. So an upfront milestone royalty sort of partnering deal. Some of these we'll keep in-house. So, we had originally thought we would probably keep PKU in-house. It's such a big opportunity and fairly straightforward for us. We already have that bug. But it's really nice to have a partner when you're a young company, and a young company can be validated by a much stronger partner. So that's kind of where we're going on that. So this is a biologic. It's a modified E. coli. It's an engineered E. coli. We're going to do a master cell bank, a working cell bank. You can ferment this. You can ferment this up to 10,000 liters. This construct with the CDMOs that I know today from being in the microbiome field, they can do an E. coli like this construct, 10,000 liters. You lyophilize it, put it in a enteric capsule, survive the stomach, and it expands after that. There's a minimum dose to colonize. So it's a method patent today that started. So you have exclusive rights to a UCSD patent that the professor came up, Professor Zarenpar at UCSD. So that's what's starting us off as a method patent. But as each engineered microbe, we will put a composition of matter around each one of those. Okay. So we're just starting with the method patent. Get us going. Somebody's calling me. Okay. Any other questions? Thank you.